In this video, I'll be describing the final stages of the virus life cycle, maturation and exit. So just a quick reminder, what we've talked about so far. The virus life cycle consists of attachment, entry, biosynthesis, and then these final stages, maturation, and then exit. So in biosynthesis, the virus has taken over the cell in order to make lots of copies of the genome and the capsid proteins. So in this picture here, just kind of showing one example, genome and capsid proteins. So these are essentially the building blocks of a virion. Remember, a virion is the final complete virus particle that can infect other cells. So the cell is filled with lots of copies of this genome and virus particle, but they have to be genome and virus proteins or capsid proteins, but they have to be assembled. And different parts might be made in different parts of the cell. In this picture here, it's showing an example of a DNA virus. Right? That genome is in the nucleus. If it's an RNA virus, that's often in the cytoplasm. But let's not really worry too much about where these pieces and parts are. Virus assembly is actually a really complicated and finely tuned process. And we don't have time to talk about a lot of the details. But the main idea, really the main take-home message here is you have these individual protein subunits kind of in the cell. And specifically, this is going to be the capsid protein. Remember, the capsid assembles around the genome to form the capsomere. And then you also have lots of copies of the genome. Again, we'll use our DNA example here where it's circular, but the same thing would apply. So here's our genome. So there are lots of copies of all of these things. And essentially these pieces interact with each other and they kind of take over the cell to direct an assembly to bring these capsid proteins around the viral genome. So usually every single virus particle has one copy of the genome. And so what has to happen is these building blocks come together to essentially form a package around. Right, so here we have our individual proteins called capsomeres. Right, those are the individual building blocks, and the whole shell together is the capsid. So that refers to the protein portion. And then all of this together, when it's assembled and mature and active, is called the virion. So sometimes this maturation is complete once inside the cells, sometimes it requires additional steps, but the main take-home message here is that you're putting the building blocks together to start to assemble the virus. So the last step is viral exit. And we're going to discuss two ways that the virus can get out of the cell: budding and lysis. So let's look at budding first. So in viral budding, the virus actually is going to push its way out of the plasma membrane. And this is something that's only going to happen with enveloped viruses. So here we have the plasma membrane of the cell. We have the cytoplasm here, and then we have the extracellular area here. So the virus capsid, the assembled capsid and genome, are going to make their way up to the plasma membrane. And so I'm just going to draw it in this more simplified, easier to draw shape here. Our capsid and our genome, they make their way up to the plasma membrane, and then they're actually going to trigger that plasma membrane to sort of push itself outward. And then that capsid gets included. And so eventually this will continue pushing itself outward until it pinches off from the cell, and we're left with our complete virus particle. So again, this is only enveloped viruses. So this would be the envelope here, and then inside is the capsid, which contains the genome. So here's a diagram showing this in a different way. They've actually kind of drawn this upside down from my picture. So here we have the capsid kind of assembling near the plasma membrane, pushing out of that plasma membrane, and eventually detaching itself. And so that means that virus is going to be surrounded by the envelope as it leaves. And the other thing that's interesting about this is part of the way the virus gets its spike proteins. So those spike proteins are actually sent to the plasma membrane so that whenever that virus buds off, those spike proteins are included. So the other way that viruses can get out of cells is through the process of lysis. This is a little bit more simple. The idea here is that these virus particles are assembling and maturing inside of the cell, and the virus really takes over. So here is an example of a cell that's infected with a virus. This is actually a plant cell that's infected with a plant virus. But I chose this picture because it really nicely shows the process of lysis. So here is kind of outlining the entire cell. And then those hexagons there are the virus. So though you can see that this cell is just completely full of virus particles. And eventually that cell wall breaks down and all of those virus particles kind of spill out of the cell. So that concludes our stages of the life cycle of the virus. So the virus attaches, it enters, it makes genome and proteins, it assembles everything into the virus, and then it has to get out. And the way that it gets out really depends on whether or not it has an envelope. So envelope viruses have to bud, and non-enveloped will get out through lysis.